Okay, so we're on chapter 9, section 4, and we're going to talk about how the glycolytic pathway is regulated. Now that we know what happens in it, how is it regulated? Well, it's enzymes, right? And so all enzyme activity is controlled by two things: substrate availability and the level of catalytic activity. And so these two factors are going to control the flux through all metabolic pathways, including glycolysis. So there are two basic categories of reactions within glycolysis. There are reactions where you have a delta G that's close to zero, whether it's positive or negative, it's just really close to zero. These are reversible. These have enzymes that are going to work at full capacity, right? So the direction of the flux doesn't depend on the enzyme, it depends on the substrate availability. Then you have reactions that are at a high delta G. So I guess I should write this: two types, right? Two types. You have a delta G close to zero, and then you have a delta G that is highly negative, right? So these highly negative ones are basically irreversible. Our delta G where we're close to zero, these are reversible. The ones that have a high negative delta G, these are irreversible, right? Irreversible. And it's because our physiological metabolic concentrations are subject to the enzymatic control, not the substrate, but the enzyme. So we have three enzymes that really fit this description, this definition of our highly negative delta G reactions. Those are hexokinase, PhosphoFructose 1, and pyruvate kinase. So these are considered the rate limiting steps within glycolysis. So you remember the little animation, and the little animation, this is where it necked down, right? And these are the rate limiting steps because we can alter the enzyme activity, right? And you can make the enzyme activity level very low, even though you have high substrate concentrations. So, and if you think about it, right, if everything were reversible, you'd be able to go through gluconeogenesis with all the same enzymes, and what would control whether you're going through glycolysis or gluconeogenesis, right? How do you determine that? Well, in this case, because we have these rate limiting steps, what happens in gluconeogenesis, right, which is the opposite, which requires ATP and is an anabolic pathway, is that we're going to utilize all the same enzymes from those reversible steps, but in the pathway where we hit those irreversible steps, we're going to have unique and specific enzymes to gluconeogenesis, and which are in these three steps, right? So we're going to talk about, in this section, basically the ways in which substrate availability and enzyme activity levels control our flux in the glycolytic pathway. So the first one we're going to talk about is PhosphoFructose kinase 1. So PFK1, right? So in PFK1, what does PFK1 do? PFK1 converts fructose 6-phosphate into fructose 1,6-bisphosphate. And this is a reaction that requires ATP. So you put ATP in, you get ADP out, right? So this is one of those enzymes that's going to regulate our flux within glycolysis. And so how do you control the enzyme? Because it's not going to be dependent on substrate. It's going to be dependent on enzymatic activity. Well, you have allosteric regulators. So there are two different kinds of allosteric regulators, and here are all of your allosteric regulators. Right, you have two different types. You have ones that are going to be positive, that are going to activate, right? And there are ones that are going to inhibit. So the activators are fructose 2,6-bisphosphate. This is a metabolite that controls carb metabolism. And so remember we said there are a lot of connections with different pathways. This is one of them. And we haven't talked about carbohydrate metabolism yet. So you just have to know it comes from there. And then AMP and ADP. So this kind of makes sense, right? Because if you have a lot of AMP and you have a lot of ADP in the cell, that means that you have a low energy charge in the cell. The cell doesn't have a whole lot of ATP hanging out. So if we can break down glucose and we can get some more ATP, that'd be great. Now, on the other hand, what are your negative inhibitors? Your negative inhibitors are ATP and citrate. So ATP is a high energy charge in the cell, right? So you've made a lot of ATP. You don't need to make any more, right? We're not going to waste it. Maybe we're going to go and use that glucose for other things. And I'll show you kind of at the end of next section, what are some other things that you use it for? And then citrate. Citrate is another inhibitor. And citrate accumulates when your citric acid cycle is inhibited. And so if you're going to inhibit citric acid cycle, you kind of want to inhibit glycolysis as well. But we'll talk about how those are all kind of interrelated once we talk about more of the pathways. So if you look at a graph of activity of the enzyme versus our substrate concentration, what you can see is depending on what allosteric regulators are present is going to control enzymatic activity. So if we have ATP, right? ATP is supposed to be our inhibitor. What are you going to do? You're going to slow down your reaction. If you add ADP and you add fructose 2,6-bisphosphate, both of those are going to activate our enzyme, and so you'll see the kinetics of the reaction speed up, right? And the whole way that these allosteric regulators work, remember we've talked about this in the past, that they're going to bind somewhere relatively close to our enzyme active site, and when they bind, they're going to do some kind of conformational change, and that conformational change is going to alter the activity of our enzyme PFK1. And so you have basically two conformations, just like that T and R state, right? Our T state is inactive. Let's say it's bound to ATP. Our R state is the active form where we're bound to AMP or ADP, right? Okay, so let's talk about another way that glycolysis is regulated. Glucokinase. Glucokinase functions basically as a glucose sensor, and we talked about this a little bit in the last section. And it acts as an insulin, it induces insulin release from our pancreatic beta cells in response to high blood sugar levels, right? So it does the same job as hexokinase. So, so here's glucokinase, right? Right here, glucokinase, and it's similar to hexokinase, right? And so the whole point of hexokinase and glucokinase, right, is converting glucose into, so we're going to convert glucose into glucose 6-phosphate, right? And so it does the same job as hexokinase, but here's the thing. It has a lower affinity for glucose, and it's not inhibited by this glucose 6-phosphate product, which means that it takes a lot of glucose in order to activate glucokinase. So why is that important? Well, glucokinase is only in our liver and our pancreas. And so you only want to activate glucokinase if you've just eaten, because this is the pathway that's going to activate insulin release. And so if we have a big enough meal, then it's going to activate our glucokinase. When our glucokinase works, we convert glucose to glucose 6-phosphate. We go through glycolysis, right? We're going to make ATP. Once we make our ATP, right, our ATP is actually going to inhibit a potassium channel. So here's our potassium channel, right? That inhibition of that potassium channel is going to actually induce membrane depolarization because of that charge difference, right? And then you're going to activate calcium channels because of this difference in charge, because they're voltage gated. You'll have this huge influx of calcium into the cell, and that influx of calcium will cause these vesicles that contain insulin to be released, right? Well, to bud, to hit the membrane and then release their contents into the bloodstream. And so then insulin can go out into the bloodstream and tell all the other cells, hey, we just ate. We need to pick up all the glucose from that's floating around in the bloodstream, right? But you really want, it's really important that we have a low affinity for glucose, because you don't want just a little bit of glucose to activate this pathway, right? Because then you would have low blood sugar, but then signaling your body to take up a lot of glucose, it doesn't make sense. It doesn't work, right? And it's also really important that it's not inhibited by the product of the enzyme here, because if it were, then as soon as it was activated, it would be shut off. So that wouldn't work either. So those are two really important regulation pathways. Okay, so I know this looks super complicated, but you don't have to memorize it. I really just want you to see how monosaccharides come into glycolysis. They can come in from a bunch of different ways. You can drink milk, right? If you drink milk, then milk has lactose inside of it. So then lactase is going to break that down. Let me see if I can zoom in. It's going to break that down into galactose and glucose. So it'll make galactose and it'll also make glucose. You can get maltose. Maltose is a plant starch. And you have enzymes like alpha amylase that are going to start to break them down, those starches into monosaccharides, which are maltose. And then the maltose can then go through and be converted into glucose through maltase, and then go through glycolysis, right? Sucrose, right? Sucrose is our table sugar. Fructose is the sugar found in fruits, right? So there are a bunch of different ways that you can enter into glycolysis. You don't have to start at glucose. You can start at all kinds of different sugars and then go into glycolysis. And this is something that you would know if you had a lactose intolerance, right? So if you have a lactose intolerance, right, you consume lactose and you don't have lactase. So the human gene for lactase is usually expressed at really high levels in infants, right, to aid in digestion of breast milk. But its expression declines as we get older. And so either as you get older, this happens no matter who you are, unless you're Scandinavian, which is sort of interesting. But anyway, as you get older, you express less of it. Or if you have a mutation, no matter how old you are, and you cannot make lactase, this will happen to you. So if you have no lactase, then the bacteria in your gut are going to use that lactase, and they're going to ferment it, and they're going to make lactate. They're going to produce H2, they're going to produce methane, right? They're going to make gas. So you're going to feel really, really gassy. And then depending on how much lactose is there, if you have a lot of unhydrolyzed lactase, lactose, not lactase, if you have a lot of lactase, gosh, I said it again wrong. If you have a lot of unhydrolyzed lactose, the sugar, then you have a high concentration outside of the cells. And so water is going to move from your intestinal cells into the lumen of your intestine, right? To kind of basically equalize the concentration of solutes inside and outside the cell. And so you'll end up having diarrhea, right? And so those are the symptoms of being lactose intolerant. You can take a supplemental pill that has the enzyme lactase in it before you eat, or manufacturers produce milk where they've added lactase to the milk, and so it gets hydrolyzed before you even consume it. So either way works, right? And you have all kinds of different ways that you can enter into the pathway, and it doesn't necessarily even have to be sugar, right? You can actually have glycerol coming in, and all kinds of different things. And we'll talk about more of these integrations in the pathways once we get through more of the pathways. So lactose is kind of interesting because you're going to make, when you break down lactose, right? When you break down lactose, you're going to make two things. You're going to make glucose here, and you're going to make galactose, right? So glucose goes straight into glycolysis, but you have galactose here. And galactose is kind of funky because you can't use it straight away inside of glycolysis. So you have to convert it. You have to convert it to glucose first. So you take your galactose and it goes through galactokinase. Galactokinase, it's a kinase, right? It's going to add a phosphate group. So we get galactose 1-phosphate. So this conversion is going to take actually 4 steps in order to go from galactose to glucose. So we have our galactose 1-phosphate. Now, that galactose 1-phosphate is added to what's called UDP, uridine diphosphate glucose, right? So our uridine diphosphate glucose basically converts, right, into a glucose 1-phosphate molecule and then a uridine diphosphate galactose. So it basically swaps the sugars, right? Now that we have glucose 1-phosphate, we can convert that to glucose 6-phosphate with a mutase, right? And then it can enter the glycolytic pathway. But you got to remember that this UDP galactose has to be converted back to the UDP glucose in order to keep going through this cycle. And so we can go again and again and again, right? But that's how you get some of these sugars into the glycolytic pathway without them being glucose. So our glycolytic intermediates provide carbon backbones for all kinds of different anabolic pathways and regulatory molecules. So what you see right in this blue shaded area here, the blue shaded, right, that's glycolysis. So what you see coming out of it are those outflow of metabolites from glycolysis, right? So glucose can leave, right, and enter the pentose phosphate pathway, and we can actually go and make nucleotides, which is really, really important. But this outflow is going to decrease our substrate availability, and you can actually have inhibitory effects on the pathway flux. And so it's important to understand that glycolysis isn't just a straight pathway, right? It's not the only thing that can happen within the cell. You can go and do different things. You can say, okay, I've got a lot of glucose, I've eaten, but what am I going to do with that extra glucose? Well, I'm going to shuttle it out as glucose 1-phosphate, and I'm going to go store it as glycogen in my muscles, in my liver, right? There are all kinds of different things that can happen if you have too much glucose. If you have too much glucose, you could actually shuttle it out at a later point and make triglycerides and make fats, right? So there are all kinds of things that are really, really important that are provided through the glycolytic intermediates, okay? All right, so that is the end of 9.4.